Chapter 16, Preparation for an Adaptive Immune Response. The learning objectives for this topic are: describe the two classes of major histocompatibility complex, or MHC, proteins with regard to their location and function. Explain the roles of antigen-presenting cells, e.g., dendritic cells and macrophages, and MHC molecules in the antigen processing and presentation. Contrast endogenous antigen processing with exogenous antigen processing. In this topic, we will be covering the roles of the major histocompatibility complex and antigen processing. First up, roles of the major histocompatibility complex, or MHC. All cells have what is called MHC class 1. And basically it holds and positions epitopes for presentation to T cells. Well, what does that mean? Well, we're going to be talking about that in the next two slides. But basically, if you look at this looks an awful lot like a T cell receptor. And you'll notice there's an antigen binding groove. And the purpose of this is to display antigens that come from inside the cell. Now, like I said, you find MHC1 on all cells. MHC2 is a variation on MHC1 that we find in professional antigen-presenting cells. What do I mean by professional antigen-presenting cells? These are cells that present antigen to T cells on a regular basis. So that would be dendritic cells, which are a type of macrophage, and other types of macrophages. We also find them on T cells, and we also find them on B cells. So in other words, you find MHC2 on your adaptive immune cells. Now, on to how they present antigen. We're going to talk first about processing endogenous antigen. Now, remember from previous recorded lectures, we talked about endogenous antigen being those antigens that come from inside of a cell. Let's say that this set of cells are skin cells. They look like epithelial cells, so why not? So epithelial cells make all sorts of proteins, but we're going to say that these guys are differentiating into epidermis cells, and they're starting to produce keratin. Keratin is a protein, it's a polypeptide. So in the process of making these polypeptides, some of them are pulled off and chopped up into epitopes. Think of it as on the factory lines. They pull off some of the products so that we can make sure that they're being made properly. That's kind of what's happening inside of the cell. So these epitopes are put into the lumen of the endoplasmic reticulum, where MHC1 proteins are embedded in the membrane. So we take these epitopes, we stick them into the MHC1, and they're pinched off into a vesicle, and this vesicle is taken to the surface of the cell where it displays it. So basically what's happening with MHC1 is the cell is displaying pieces of whatever it's making and saying, this is what I'm making. T cells and natural killer cells come along and check these out to make sure that everything is okie dokie. If they're making what they're supposed to, they're left alone. Because, as you may remember, T cells that recognize self-antigens or antigens that your body's supposed to be making are killed during the development process. If, suppose, this is infected with a virus and it's displaying viral proteins, it can be recognized as being an infected cell, and T cells and natural killer cells kill them. Let's move on to processing exogenous antigens, or antigens that come from outside of the cell. In this case, the exogenous antigen is coming from a pathogen that's been phagocytized by an antigen-presenting cell. So in this case, it's a macrophage. So it phagocytizes this poor little bacterium, and we talked about phagocytosis and how the cells are chopped up, which results in epitopes in the phagolysosome. Well, we've got vesicles that have been pinched off from the endoplasmic reticulum, just like with MHC1s, and they're in a vesicle that fuses with the phagolysosome. In the process of fusing, these exogenous antigens, or antigens that have come from a non-human cell, are placed into the MHC2 protein, and this vesicle fuses with the cytoplasmic membrane, and MHC2 displays the exogenous antigen on the surface of the cell. Now the antigen-presenting cell can go present this antigen to T cells and B cells. That's it for this topic. Nice short topic to make up for the very long topic previously. And here are reminders of what you need to learn from this topic.